it's my pleasure to introduce Dr. Stephen Balin. Dr. Balin's the Virginia and D.K. Ludwig Professor for Cancer Research and a Professor of Oncology and Medicine at Johns Hopkins University, as well as
a great human being in his own right.
and Bob says a great human being in his own right, and the Deputy Director of the Stanley Kimmel Comprehensive Cancer Center at Johns Hopkins University. The title of his talk is Cell Signaling and Clues to the Origin of the Cancer Epigenome.
Yes, I'm trusted to move this. Let's move that. Kill somebody, including myself. Thank you. Let's just move this back there. There we go. I'm going to take this. Well, thank you very much. Not ever having been fed badly in this building, it's a pleasure to be here, and I thank the organizers terrifically for inviting me. And in many ways, I'm going to continue the same theme that you've heard, and Bob mentioned it several times, and sort of push the epigenetic component of this, but this type of plasticity, this type of movement, and how epigenetics plays into it is very much the theme that I'm going to continue. And so just to set the epigenome in a very brief cartoon, everybody in this room recognizes from ENCODE all the studies and the deep sequencing that's gone in, how complex the epigenome is. During our time in my group working on this, we've been very promoter-centric and worked on changes that have altered gene expression abnormally through promoter chromatin and DNA methylation changes. But now in this cartoon from Peter Jones, my colleague in this review, we know that all the chromatin regulation that takes place here in control of DNA methylation is totally germane to how insulators work for boundaries in the epigenome, for how enhancers work and how they're regulated, and particularly how non-coding regions, microRNAs, and long non-coding RNAs that are so critical play a role. So we really have to think about this whole context, even though I'm going to focus here. We have abnormalities that can occur in any of these places, and we're probably at the tip of the iceberg in really cataloging those and knowing how they change tumor cells. So the whole key to epigenetics is obviously a packaging of our DNA around the nucleosome and then modifying the histones that make up the nucleosome. We have a whole series of proteins, as you know, that for the histone modifications, put them on or write them, take them off and erase them, read them, and particularly those that move the nucleosome around. So all of this is really the regulation. I'm going to talk about things that balance DNA methylation versus a long-term silencing system of polycomb, but again, this complexity. And I'll just put up something. I'm not going to talk about the translational in depth, but I put the targets up indicating that all the pharmaceutical companies, Constellation here in Boston, all of the people working have programs, most of them, for targeting each of these individual proteins that regulate this epigenome, including DNA methylation, and more thought now be given to the proteins that move the nucleosomes as well as means of cancer management possibilities. And many new drugs are coming into the clinic, actually being tested in phase 1 now, that modulate various aspects of this. So it's translationally extremely important. And we also have to remember now that there's a very high frequency of mutations turning up in virtually every cancer type that encode for the proteins themselves that control this epigenome. And one of the thoughts that's coming about, as I'll stress, by the genetics community in cancer research and epigenetics is it's very difficult to reach in and repair the mutation. But the epigenetic phenotypes, which we really need to understand more and more for these mutations, are much more reversible. So the hope might be you can come in downstream from the mutation and make manipulations that will be translationally, again, very important. Now, we've worked for many years, as I said, around the promoter on a shift in cancer cells which occur, probably in the very same cancer cell, losing DNA methylation at CPG sites like lollipops, and gaining methylation in CPG island promoters that are ostensibly anywhere during development to adulthood, except as you begin to age, don't take on methylation in these regions. Hundreds of genes in every single patient's cancer can take on this change. Obviously, we don't know which of all those genes are the most important. Some of them are critically driver genes, von Hippel-Lindau, P16. Others are more nuanced, like in the Wnt pathway. Bob mentioned one of them that frequently gets methylated, or two of them, is SFRP1 and DKK in many tumor types, and there are many more we don't know about. But suffice it to say, this is a very frequent change in cancers that we've worked with. And epigenetics really is all about, in my mind, is boundaries. And so if you have a region that shouldn't have DNA methylation or more heterochromatic change here, what you're really doing is setting up boundaries where the machinery that catalyzes DNA methylation in protein complexes is excluded from that. And what I'm going to stress today is that we think there are big protein complexes that hang out for DNA methylation and repression on either side of a protected region like this, and that one of the keys is cell stress. Stress for cell survival that may be something inherent to what Bob was talking about that cause shifts of these proteins to occur into these regions. And retention of the effects of these proteins may be very much about setting up some of these epigenetic abnormalities that I'm talking about. And I'm going to talk more about this. But this stress concept is what I want to really focus on today. And what I'm going to suggest, and again, hopefully some being right and data-driven, but hypotheses at point, that one of the big stimuli to the abnormal epigenetic changes that occur in promoters and other regions, probably in cancer, is a survival mechanism. Cells under stress, particularly in cancer risk traits, inflammation, reactive oxygen species, that to survive are adopting a cellular state, and that state is really like EMT. It's more a stem or progenitor type state with a chromatin inherent to it that is really very susceptible to a molecular progression of epigenetic abnormalities that can more and more stably silence genes and probably in some instances may switch to activation of genes. And there's a bias in these abnormalities in genes fundamental to the control of development pathways and certain signaling pathways. And that transcription factors, DNA binding proteins, Wnt and other stem controllers you've heard some about are very much susceptible to alterations of this type. And as I just showed you, there are key intersections with genetic abnormalities. So I'm going to show you a bit of our work on this that's been spearheaded over the past several years by Harry Eswan in our group. Sarah Johnstone did some, who used to be a graduate student in Rick Young's lab, and bioinformaticist Leander Van Ness. Particularly work on inflammation and stress and some of the protein interactions that have been done by Heather O'Hagan and Yi Kai in the lab, informatics from Wei Wang, and some of the biochemistry from Subhajit Sen. And I'm going to show you what this group has been working on. So our concept is that there may be a molecular progression here during the evolution of cancer from risk states to frank neoplasms. And some years ago, our lab and Peter Laird's lab and Howard Ceder's lab, sort of simultaneously in different ways in colon cancer, came to the recognition that maybe half or more of those genes with that DNA hypermethylation that I just showed you that happened in colon cancer were, by history of chromatin, marked by polycomb, the long-term silencing group of proteins. It puts on the repressive mark, H3K27 trimethylation in normal development. And that this bias towards these proteins or these marks here reviewed by Howard Ceder, that what could happen during cancer is that you could move into a state where, say, overdrive of polycomb without DNA methylation could lead to a abnormal repression of such genes. But that if over progression in time, that the protein complexes I'll be mentioning could drive this vulnerable state to DNA methylation, which is a more stable silencing state and much harder to revert. And along the lines that Bob was going to talk about talking about in cell population, I think tumors during progression could be caught anywhere in here for different patterns of genes that might have taken on these driving changes during tumorigenesis. And this is the concept, again, that I'm going to be banging on here. So one way that we have taken this further than just the colon cancer cells, and we and other groups have been studying this, is to look at a mature system from which bone tumors ostensibly come, being mesenchymal stem cells that can be converted experimentally to osteoblasts. And we took human mesenchymal bone marrow stem cells that could do this and did a probe for a mark called bivalency, which is not only having at the promoter of these genes the H3K27 mark, but as Brad Bernstein is, many of these genes can sit in a state of bivalency where they also actively have the active H3K4 trimethylation mark. And that's a poised state in this concept of bivalency, where the genes would be held in a state that is in low transcription. They're poised. When the signals come in, that gene will either convert, depending on the lineage commitment, to an active gene and take on the H3K4 trimethyl mark and be active, or can stay in the polycomb mark and have that as a sole repressive mark. And the key to this is none of these bivalent genes, virtually none, are controlled normally with DNA methylation in the promoter region, and that's a key to this concept. So what we did here is take a bone tumor, a culture osteosarcoma, compare it to this, do a ChIP-seq approach to the bivalent mark, across the genome DNA methylation probe, and simultaneous gene expression. And if you look at these osteosarcomas in this heat map, which is an infinium array for going across the genome, and this is biased only to probes in the CPG islands of promoters, you can see about 380 genes, as shown in the red here, that in the bone tumor take on DNA methylation in these regions, that the mesenchymal stem cells, the osteoblasts, and embryonic stem cells, any way you compare this, this is a de novo cancer-specific DNA methylation. And if you examine the history of those genes in our paradigm, or go back to embryonic stem cells, about 3/4 of these genes in embryonic stem cells will carry that bivalent mark, both the H3K27 and the H3K4. But what happens to this bivalent mark normally, and here's our own data from the mesenchymal stem cells and the osteoblasts, and here's data from databases. If you trace the history of these genes back, as I said, the orange bar indicates here that for the osteosarcomas, but you can do this for gastric, lung, leukemias, the orange bar is bivalency. We find about 70 to 80% of these genes trace back to stem cells, not only embryonic stem cells, but hematopoietic stem cells as well, as being bivalent during that stem state. And then what happens during normal commitment is that you can see here the mesenchymal stem cells convert those genes to maybe half of them in the osteosarcoma, which I think is here, active, and half being polycomb alone, and very few that are still bivalent. And you can see this in the hematopoietic system as well across differentiation. So this is what normally should happen. This plasticity and this conversion should occur. No DNA methylation, no locking in, if you will, other than the polycomb system or the H3K4 mark. And most of the genes, as I said, that you're seeing in the red right here should be controlled that way. So who are those genes? Well, it's really tremendously of interest, to me at least. The ones that are normally retain polycomb and are bivalent and get silenced with the DNA methylation are mostly developmental transcription factors, genes that are key to the regulation between remaining in a self-renewal compartment or committing to lineage or differentiation. These are the ones that are the polycomb mark genes in the osteoblast and the mesenchymal cells and other. So these are really a developmental program. Interestingly, those that convert to the active mark in the progenitors or have an active mark in the embryonic stem cell, H3K4 genes, are mostly cell signaling genes, genes that have to do to cell surface receptors, neuronal development, the projections that neuronal cells put forth and whatnot. So there's a real difference in the body of genes depending on whether they went active or they remain more repressive in a stem and progenitor state. Now, some years ago, Bob Weinberg's lab at the transcript level described features, partly which were epigenetic, of the more aggressive tumors that were more embryonic or stem cell-like, and then Stu Orkin, using protein networks, pushed this theme to what he called the Orkin module or polycomb module from protein interactions. And what he stressed is that in tumors, you don't see much change in the core embryonic factors. MYC module is always upregulated. And then there was this polycomb module he defined that ought to go up during differentiation, but the targets of the polycomb stayed down. And what we looked at is, are there components of this so-called Orkin module polycomb that actually get DNA hypermethylated, go on to that more stable change that I talked about. And indeed, we found that in multiple tumor types, about maybe 20% on the average of the so-called Orkin module polycomb genes actually took on DNA hypermethylation. And the pattern's different for different tumors, and the numbers of the genes that we found in the genomic survey are different for different. But the point for expression is illustrated here. If you remained bivalent for these genes in the tumor cell, for instance, you had a relatively low transcription. If those bivalent genes took on the DNA methylation, the expression of all those genes on the average is much lower. If it was an H3K4 alone gene and took on DNA methylation, of course now the delta is really big because that would be an active gene in the stem and progenitor cell normally, and with the methylation is inactive. So there's a real expression consequence, we think, for the degree and the change in those chromatin states as tumors progress. And sort of our summary, here's what we're hypothesizing, conceiving, that normally there's many of the genes that get DNA hypermethylated in cancer should be controlled in a plasticity sort of way by bivalency. And during tumor progression, there are a series of events that can occur where those genes are very vulnerable to replace this type of chromatin with DNA hypermethylation in these promoters, and they would have lower expression. And again, there would, along this progression, different tumors, different patients' tumors could be anywhere in this progression in the pattern of genes that they have that have manifested these changes. And that this could be one way, to a different degree, whereby, say, polycomb or DNA methylation could help lock in maybe that more EMT state and make it a little more rigid and more difficult to convert back to the more committed state. But there would be a room for plasticity in here, particularly if it were polycomb alone and not DNA methylation that did this. We have some evidence for this, of how it can work with genes that are involved. A number of years ago, we cloned one of these developmental genes. It turns out to be one of these genes frequently hypermethylated, never mutated or rarely called HIC1, and made a knockout mouse for this transcription factor, which is a developmental gene. It's embryonic lethal in the homozygous knockout state. But the heterozygotes get a stem expansion. And you can see it here in the crypt of this. There's the wild-type mouse with the number of crypts outlined here for beta-catenin, pervilis, and you can see increased numbers of crypts. And this animal is tumor-prone. Match it with APC, and you get enhanced tumorigenicity. Recently, a mutation, there's 3 papers that came out, Craig Thompson's group and others have described IDH mutations that occur in brain tumors, and it's a complicated metabolism story, but at the end of the day, these IDH mutations take on hypermethylation of many of these polycomb bivalent genes. And they found that if you engineer cells for this IDH mutation, or you look at natural gliomas that harbor this mutation, their phenotypic manifestation is a freezing in a stem-like state, which is reduced capacity to drive to differentiation like neural. But if you demethylate these genes, and there's no proof yet in my mind that this is the specific step, but you do demethylate, you can restore more inducibility to differentiation. So I'm just pointing out some data-driven possibilities here that this may not be a wrong concept. Now, I'd like to make the postulation that the state of this chromatin, and Bob mentioned this as well, could be determined by the compartment in the epithelial renewal system from whence the tumor emerges, maybe the rapidity of tumor emergence, because I'm going to show you we think the DNA methylation takes time to develop. And it will reflect the pattern of the chromatin of that tumor. And I think this is going to be a potentially translationally and very more important concept. And I want to show you a couple of illustrations that suggest this. This is that Infinium heat map that I showed you. On this side, the yellow means these are all promoter CPG island genes Almost all of them that are in the yellow, they're methylated, are polycomb target genes of the type we've been talking about. These are adenocarcinomas, non-small cell of the lung, and data that's being revised for the TCGA, a project on lung adenocarcinoma. But what I want to point out here is you have groups of these tumors where you have infrequent methylation in these genes, intermediate, and then you have this so-called hypermethylator phenotype of these polycomb genes, or so-called CIMP. Breast cancer is really interesting in this, to my mind. Several groups, these are our data, but others have shown this. So this is that same Infinium heat map. Now, red is hypermethylation of these CPG island promoter regions. The red is Chuck Peru's signature here for basal or triple negative tumors. The gray and the black are luminal tumors, the black being, I think, luminal B and the gray. But what you can see is a stark difference in these genes, much more hypermethylation in the luminal tumors than in these basal or triple negative. And several groups have hypothesized that the drive here for downregulating the polycomb genes is polycomb itself, overactivity of the enzyme that catalyzes that mark, EZH2, where these genes in the luminal take on this different change in DNA methylation. And I raised the question, with targets now being inhibition of EZH2, agents out for reversing DNA methylation, do we have to know the chromatin set of the gene if we're going to do so-called epigenetic therapy? Would you be approaching these tumors one way and these tumors a different way? And it's a question I think that we'll face in the field. Now, in the final part, how do these changes come about? I've said stress induces this. And I'll show you the data and the systems we're building for this to do this. So we have a system which is industrial. We apply reactive oxygen species. We apply H2O2 to cells in millimolar quantities to raise their ROS levels and look just 30 minutes later for changes that occur in the DNA methylation machinery and associated proteins. And I'll show you more physiologic, if you will, pathophysiology in a moment. But these are embryonic carcinoma cells that we've used. And we have found a complex where we can co-immunoprecipitate DNA methyltransferases that catalyze DNA methylation associated with polycomb genes like EZH2 and other members of the complex, a key histone stress-induced deacetylase, SIRT1, and HDAX. And the key here is that during this stress, within 30 minutes, the major maintenance protein for DNA methylation tightens to chromatin. Normally, if you do a salt gradient, a little more already tight in stem cells, very little would you see in an epithelial cell here. And after 30 minutes of treatment, you see on this salt gradient, particularly with a high salt here, this DNA MT1 tightens to chromatin. Along with it, you get SIRT1 moving to the nucleus and tightening to chromatin. And if you do a glycerol gradient and look at these proteins in the complex, you see two things. The complex size with treatment enlarges, so this is larger size of the complex going this way, and you can see these by Western. Most of these are absent in this size range here, present here. You can co-immunoprecipitate them right out from these fractions. So there's tightening to chromatin and enlarging of this complex for polycomb and DNA methyltransferase proteins. If you look along chromosomes, you can see what I think is a fascinating movement. This is, again, within 30 minutes. So we're tiling along chromosome 21 here. Here's chromosome 19 here. And what you see is coordinate movement, including this DNA methyltransferase, this tighten away from GC poor regions into, and this is telomeric on chromosome 21, GC rich regions. And on this chromosome, this is where the transcriptome is enriched. This is where you get CPG islands. And so you see this really rapid switch. The downtick means the proteins are leaving, and they co-localize with the H2X phosphorylation, in this case, that would go with the DNA damage. And wherever those regions are on different chromosomes, you'll see the switch into the CG or GC-rich regions. And this is 8-guanosine, which would be the damage product at the MYC gene and the actin gene. And this is where you see these complexes localized. And it happens to all CPG island genes. It's not just genes that get repressed. It can be MYC. And I'll show you why we think this is happening. And there's real functional consequences to this. Within that 30 minutes, you drop the active mark H3K4 in general, particularly at the high expression CPG island genes, you tend to raise the silencing mark of H3K27 and the deacetylation mark that SIRT1 would cause here. And you can see this across the genome. You can see this at MYC. Here's a silencing mark going up, the active mark going down. And ostensibly, every CPG island we look at takes on this change. And during that 30 minutes, if you do nascent RNA transcription, these genes are shutting down. And we think maybe this is a mechanism by which during that DNA damage you want to shut down transcription and not try to repair through a transcribing gene. And we have some evidence building that that may be the case. Another fascinating change, which I did not think would be possible, and the postdoc and graduate student working on this convinced me by sequencing that it was, that for the low expression bivalent genes that we're talking about, if in this particular cell they weren't DNA methylated, almost as a function to how low their basal transcription was by sequencing, so we're sequencing CPG methylation here, you would begin to see in that CPG island an increase in CPG methylation. So even within that short period of time, we could see this. And if you look at how this looks in the shifts right at the promoters, which I think is interesting, there's some really important changes. So what we're looking at now is the position of one of the DNA methyltransferases for de novo methylation 3B, and we're looking at some of the genes you heard about. We're looking at SFRP1 and SFRP4. Before the DNA damage in the orange, you see the peak of 3B. Here's the CPG island right around the transcription start site. The peak for the proteins on this side and this side, but not in the island. The 30 minutes of damage, it moves in. And you can see it in this gene. And here's an island that's upstream of the gene, takes on the same dynamics. And that's where all these components seem to hang out basally and shift with damage. There's one exception. If the gene is not DNA methylated, so in this cancer cell where we're working with SFRP4, which is in that family of anti-Wnt genes you heard about, is not DNA methylated. So it shows the changes that I showed you. DNMT1, for instance, is dipped here. It's dipped in the island. It goes up with damage. But if the gene is already DNA hypermethylated, and SFRP1 and 2 are in this cancer cell, then you see a complete conversion where now the DNMT1 is already in the island. And it's in the island, you can't see it as well right here as well. Still goes up with damage, but you've seen a shift in a retention of the major enzyme that perpetuates DNA methylation once it starts to get set up. Finally, to end, we have taken this to more models that are more mimic of cancer risk states. In Heather's first work on this, she actually was able to use a model where you give a bacterial toxin to a mouse. And then within 48 hours, you get inflammation in the distal but not the proximal colon. So the mouse actually serves almost as its own control. And I won't go through all the data, but what she could see by chip analyses in this very early time period was an increase of polycomb at many of these genes and some of the same types of changes which I've just been describing to you in this more subtle situation of early inflammation. What's been exciting to work that's now going on that I'll end with here is actually smoking, giving cigarette extract chronically in lower doses, letting the cells live, to human, either immortalized, and now we're doing it with a stem-like culture of human bronchial stem cells. And so these cells grow, they live indefinitely, but very quickly with the cigarette smoke, they make changes, and so far we're doing this at candidate genes. The ChIP-seq data are now all going. But what I want to show you is by ratios here, within 10 days, the EZH2 that sets that H3K27 mark is going up in these 3 genes, which all tend to get hypermethylated in cancer, low transcription. The high transcription genes, CPG island like actin, don't take this change, and this is a CPG poor gene here. And you can see that this change taking place for the first month. But what's key, if our molecular progression hypothesis is right, at 3 months, now it's DNA methyltransferase 1 that dominates it. Each of these 3 genes, and EZH2 is much lower, and that persists at 4.5 and 6 months. So that conversion, potentially that would lead to the methylation in a time-dependent way, is occurring in this model, which is interesting. And actually, we've done genome-wide DNA methylation, and here's 6 months, over 6 months without treatment. And you can see in this splay of the DNA methylation in this low expression range here, we're already seeing a huge number of DNA methylation changes that are occurring along the time spectrum that I'm showing you. So I'm going to end. I have some data on NERD to tie this together, but I'm going to end with just going through this hypothesis in summary form. What we're speculating is that in these CPG island genes particularly, you have these complexes of protein, and we've tied the NERD complex, which is very interesting for a chromatin remodeler, to this, on either side of the CPG island, and you've got barriers which we need to understand here by which they don't move into the island. But during stress or inflammation, you can transiently move these complexes in. Now, if it's cells are going to recover from that damage, it's going to be a malleable state. They're going to flip back and forth. You don't want them to stay this way. But during chronic stress particularly, and for these bivalent low transcription genes, we're postulating that when this movement occurs, that they are more prone to retain elements of this complex and move, for instance, from polycomb to a DNA hypermethylated state. And this is our hypothesis. And again, time dependency for tumor development and the compartment of which tumors come out of may be telling us something of where this progression is once you see that frank neoplasm formed. And I'll stop there, and I thank you.